in space, how experiments with fruit flies at the International Space Center are helping human cardiovascular treatment. It's a mouthful, and it's very important. So DMF Talks is the Foundation's unique version of TED Talks. We draw our speakers from an inspiring base of creative, intellectual, and scientific leaders who live locally. For the past 8 years, DMF Talks is aimed to entertain, inform, inspire the Del Mar community through this series of free presentations. A few tips for some of you that aren't so expert at Zoom so far in the last couple of months. To best enjoy our talk, we encourage you to switch to speaker view rather than gallery view. I encourage you to turn off your video so we know we have enough bandwidth for the program. I promise you'll still be able to see the presentation if you don't have your video on. We ask you to stay muted throughout the talk and direct all the questions or comments through the chat feature, which should be at the bottom of most of your screens. There'll be a short question and answer session following the talk, and I'll moderate the chat box and give the questions to Dr. Ocorr. Thank you. So Dr. Karen Ocorr earned her PhD in neuroscience from Wesleyan University after receiving her BA at Lehigh University. She was awarded an NIH postdoctoral fellowship at the University of Texas Medical School, Houston, and at Sanford University. In 2004, she accepted a position at Sanford Burnham Prebys Medical Discovery Institute. To date, she has published 68 peer-reviewed studies on heart disease using both the fruit fly and zebrafish cardiac models. She collaborates with NASA scientists and is currently serving on the board of the American Society for Space and Gravitational Research. At Sanford Burnham Prebys, she is an assistant professor in the Neuroscience and Aging Center. This evening, Dr. Ocorr will talk about her experiments with fruit flies conducted aboard NASA's International Space Station. And now we will turn it over to you.
All right, I'm going to share my screen if the gods are smiling. Okay, give it a minute. Okay, hopefully everyone can see this really cool picture of a fruit fly. I am really thrilled to be able to give you some insight into the really fun experiments that we've been able to do on board the International Space Station. As I was talking a little bit earlier about, my bread and butter is to use this system to understand what's going on genetically when hearts go bad. But we had a chance to collaborate with NASA, and that's just been completely exhilarating and exciting. And I want to share some of the insights we've obtained from these studies, which also do ultimately apply to patients and heart function in people on Earth. So let's hope this works. So first of all, why worry about the effects of long-term space flight? Until now, pretty much everyone who's been into space has been there for a relatively short period of time. Most astronauts are not in space longer than, I think the longest right now is a year, but typically on the ISS it's 6 months. Before that, it would be just a few weeks. But right now, about half of the International Space Station activity is currently commercial, and the prospect is to make the entire low Earth orbit commercial in the next 10 years. NASA wants to turn over the ISS to commercial ventures. And this is one of the first things you need to know about NASA. They love their acronyms. So low Earth orbit is LEO, and no one ever says low Earth orbit at NASA. But they are basically giving up low Earth orbit, and their current activities will be more focused on the moon. But what makes long-term spaceflight more approachable right now is the fact that we've developed reusable rockets. Since SpaceX 10, the booster rockets have these, let me get a pointer here so I can show you what I'm looking at here. So these are the extendable legs on a booster rocket from SpaceX 10, one of the first ones to land back at Kennedy Space Center. And I had the privilege of being there to see this particular landing. And I have to tell you, I was thrilled when the rocket took off. We've all seen rockets take off, but every hair on my body was standing straight out when this thing came back and landed, in the words of Elon Musk, like a rocket ship should. And so this is going to make revolutionize space travel, because imagine if you had to throw away a plane every time you traveled to Europe or another state even, if you could only use it once. Air travel would be prohibitively expensive, but this now makes space travel much more within the reach of even common citizens, and hence the advent of potential space tourism. So a good reason to worry about what happens when you're in space. Other reasons are that NASA is planning to go, NASA's planning to go back to the moon. So they have 2 missions planned to circle the moon without landing prior to 2024. And in 2024, they're committed to landing the first astronauts on the moon since the Apollo sequence of missions. And they promise to have one of them a female. And this is their current Artemis program. And they plan to establish a base at the moon's south pole, where there's a large collection of minerals, rare earth metals that are quite close to the surface and can be mined and used to create a space colony. And that's one of their long-term goals. And what you're seeing here is an imagination of the moon lander that they are currently designing, that will take astronauts from a circling space station, essentially, that will circle the moon, where rockets can interact like they do now with the ISS, and people can get off and get into these landers and then navigate to the surface. This is all planned for the rest of this decade. And finally, next decade, NASA is looking forward to missions to Mars, and these will take a couple of years. So long-term space flight is really something we need to worry about now, because it's our future. So our effort began with the box. Not this box, of course, but I'm showing this because it's about the same size as the box that we got. And this box was made by a company called Nanoracks. And they teamed up with Space Florida to put forth a prize to try and encourage scientists and engineers to think about experiments that could be done in space. And they said, all right, the prize you're going to get is the cost to take this box up to the ISS for 30 days. Discuss. It cost about $60,000, but that price is coming down, as I mentioned. And so what I'm going to be telling you about is our efforts to figure out what to do with this box and how we could use it to understand the effects of spaceflight on organ systems. So in our labs, we study flies to try and understand heart disease. And there's a lot of reasons why one might want to use the fly, although it isn't immediately obvious. But flies share about 75% of the human genes. They're mostly genes that cause disease. So that makes it a good system to look at, because we can get at perhaps some of the functions of disease-causing genes. In addition, they have many of the same kinds of proteins, genes encoding proteins that we have. So for example, muscle proteins are pretty much the same in a fly as in a human. And something called ion channels, which are important to muscle and brain function, are also common between humans and flies. And you'll see an example of that in a minute. Obviously, with respect to the space program and that little box I showed you, their small size is a huge advantage. And they have a short lifespan. So a week in a fly life is about 10 years in a human life. So we can send flies into space for a relatively short period of time and mimic what would happen to humans in a much longer period. And they've been studied for over 100 years, and there are a million gazillion different genetic tools that we have at our fingertips, including one of the latest genetic technologies that won a Nobel Prize last week, around CRISPR genetic manipulation. So here's our model system. What you're seeing here is a schematic of a fruit fly. And in red is its heart. And its heart, as you can see, is in its abdomen. And down here is an actual fruit fly that's been dissected and cut open. And oh, please work. I don't know why it worked before. Oh, I know why. Wrong. Oops. I have to, sorry. I have to change my from laser to clicker, and that may take me a minute. Okay, come on. All right. This is annoying. I apologize. I should be getting access to change this laser pointer, but I can't. Any of you techies out there, feel free to chime in if you have a solution. Let me.
There it is.
Oh.
I think you have
Yeah,
to.
there, I see it, I see it. Yeah. Thank you. It does not seem to want to let me turn it off. Let's try going out and coming back in. There we go. Okay. So here what you're looking at is a dissected open fruit fly. We've taken away its brain. We've taken away all of its internal organs. And what you're left with is this linear heart tube. And like your heart, it can beat without input from your brain. And that's what you're seeing here. And if we give it enough energy sources like glucose or sucrose, these hearts will beat for 9, 10 hours, just as you're seeing it here. And so this allows us to do a number of things to study how this heart functions. And the way we do it is by taking higher magnification movies at very high speed with high resolution so that we can get very accurate distance measurements and timing measurements for the contractions of the heart that you're seeing here. So what you're looking at are these two chambers in the middle of the heart, and there's some support cells that line the heart that's beating, which filter out proteins. They act a little bit like kidneys. So we take these movies at very high speeds, and then we analyze them. And one of the ways we do it is we can take a digital slice through our images, and we can line these up like a picket fence to get a single snapshot of the entire movie. And this is something your doctors do when you have an echocardiogram. They will do exactly this procedure. They call it the motion mode or M mode to get an idea of how well your heart is contracting. And obviously from this, you can see that we can get information about when the heart's relaxed and when the heart contracts, and get an image or an impression over the entire time of this movie that this heart's beating pretty regularly. So one of the first things I did was, one of the professors that I collaborate with at UCSD had an interesting line of mutants that he didn't find any function for his studies, but he gave me the fly. And this fly had a mutation in an ion channel, and the ion channel is called KCNQ. And what you're looking at here is an M mode from a fly heart that has a mutation in this ion channel. And here's what it looks like in real time. And I hope you can appreciate that this heart is dramatically fibrillating, the kind of fibrillation that your heart cells do if you have an area of heart tissue that is in atrial fibrillation, for example. Now, obviously, a vertebrate wouldn't be able to live very long with this kind of malfunctioning heart, but flies get oxygen through another system, not through their blood flow. And so this allows us to maintain flies that have these really badly functioning hearts and study them to figure out what the ramifications are of this kind of really awful electrical activity. And I should say that mutations in this channel are quite often the reason that you hear about young athletes in college basketball or swimmers having a fatal or debilitating cardiac arrhythmia in the middle of a game and they fall down because they had an undiagnosed mutation and suddenly under stress, their heart started fibrillating, just like this fly heart. So here's the protocol, the experiment we devised that uses this box we got from NanoRacks. Now in NASA speak, again, in acronyms, this is known as a VFB, which makes it sound very technical and official, but VFB stands for vented fly box. So very basic, just sounds better. And what we did was we put 15 vials of flies, the normal vials we keep them in the lab, into this box. Each vial had 10 females and 5 males. So in our general experience, not too many flies. And those were flown to the ISS. It took about a day after we put them on the SpaceX rocket, Falcon. And they went up to the ISS where those parents gave birth, laid eggs, gave birth to embryos, larvae, they became pupa and hatched as adults while aboard the ISS over this 30-day period. They then came back and this is one of the ironies, I guess, of current spaceflight. We've gone back to landing in the ocean, but fortunately they land for the most part off the coast of LA. So it was very close to us. And that meant it took only a couple days by boat and a little bit of time to drive down from LA to get the box back in the lab. So not too much reacclimation to Earth gravity. And this is the box when it arrived back in our lab and we opened it up. And it was full of about 400 adult flies and plenty of other embryos and larvae. One thing I should point out here, and this is the disconnect at NASA that you run into between biologists and engineers. The engineers were afraid there's little screws, I don't know if you can see them, but they're holding these plexiglass plates that fix the wire mesh over the holes so that air can go in and out. That's the vent. So those little screws, the engineers were worried would somehow come unscrewed while it was aboard the ISS. And so they wanted to put that orange tape that you see here over the entire sets, all the vents, just cover them right up. Until we pointed out to them that the reason the vents were there were so that the flies could get oxygen. And if they did that, that defeated the purpose of the vents. So we convinced them it was okay to only cover over those screws with their tape. So we get our flies back in the lab, and this is essentially what we got out. And you can almost see the KCNQ here. We sent up a couple different ion channel mutant flies. We sent up wild type flies, and we got them back. And even before we had finished analyzing all of them, we were encouraged by NASA to write a full-blown NASA grant to get money to do more of these experiments. So we were able, and you'll hear more about it, we were able to do a second iteration of this flight, but this time we were able to do it with 9 of these VFBs, which got upgraded, as you can see here. And we got an entire CTB, which is a cargo transfer bag, got an entire CTB to go up to the ISS. And we also got an entire lab at Kennedy Space Center. This is me and my technician and the Kennedy Space Center technicians working on loading flies into these boxes before our second flight. And this turned out to be just absolutely fantastic because some of the things I'm going to tell you about that we learned in the first flight, we were able to test on this second flight. So here's what happens when they get to the ISS. This is the Falcon capsule, which offloads the CTB onto the ISS. And then a number of those boxes that you saw in the lab were then placed into an incubator, a small incubator aboard the ISS. And then the remainder, we had some spares that were backups that we left just in that CTB on the shelves in the general cargo area of the ISS. And it turned out to be a really good thing we did that, because you may not know this, but the ISS is really dirty. There's mold and stuff everywhere. And all those beautiful boxes that went up and were nicely humidified and temperature controlled for a month came back, and there was massive amounts of green mold that had invaded into the vials from the air in the ISS inside that incubator. And unfortunately, we were not able to use most of the flies from those boxes. But the VFBs that were out on this shelf were actually fine, and we got hundreds and hundreds of flies from them. And so we're able to do our studies anyway. And NASA learned a good piece of information about using this incubator for things like fly culture or cell culture on the ISS. So here's what we found when they came down. And I still watch this movie, and it astonishes me. So here you've got ground control flies on the left and space-flown flies on the right. And we do a climbing assay. They will climb against gravity just naturally. And you can see those ground control flies are climbing quite nicely to the top. These space-flown flies, they get partway up and they completely stop moving. Some flies don't climb at all. And I assure you, the movie here is still running. If we had better resolution, they're moving their wings a little bit, but you can see the ground control flies are still kind of milling around on the top, and they're not holding still, but these guys just completely stopped. I've never seen that before, and it suggested to us that there's some metabolic or structural muscle problems going on. And so we did a bunch of experiments to try and get at that. So one thing we can do is we can take those fly hearts that I showed you, and we can stain them for different proteins. So here's actin, a muscle protein that you have in your muscles. And all those little red dots are collections of that muscle protein. And when we do that, one of the first things that we noticed is that the ground control flies were significantly bigger than the same genetic background flies that had been born and raised in space. So we noticed immediately a difference in size. And this has some application to astronauts, because they, when they come back, it's been recorded that their hearts become more round and a little bit less contractile as a result of being on the ISS. And our results showed pretty much the same thing. So here's a little bit higher mag of that ground control fly heart, and you can see these myofibrils, they're called, that contain these muscle proteins are arranged in a very packed way. Think of it a bit as like putting rubber bands around a tube of toothpaste, which you can see down here as a schematic. And so when those muscles shorten, you can imagine that they contract, and you would squeeze the toothpaste out of your tube or the blood out of the heart. But in the space-flown flies, you can see these myofibrils are disoriented. They're not in any really regular configuration. And that actually, in ways that we've been able to measure, caused a real reduction in the contractility of the fly heart, simply as a result of this rearrangement of these muscle fibers. So that's one thing we learned. We learned another thing, and that is that, and this was surprising, all heart structures, including yours, have what's called an extracellular matrix support. It's collagens around the heart, and also collagens that form the valves of the heart where the muscles attach. Muscles have to attach to something in order to be able to contract and exert force. In the case of fly hearts, they have this really elaborate, beautiful collagen network that surrounds the heart and actually comes in and binds to, as we've learned, binds to particular proteins within the muscle cells. And I hope you can appreciate that the ground controls here have a lot more green than those space-flown flies. And if you put the muscle and the heart tissue together, what you might be able to see is where you have the most disorganization in your muscle fiber structure, you have the least amount of collagen, whereas it's pretty much consistently interacting with the heart muscle along the entire ground control tube. And this was surprising to us because normally when hearts are damaged in humans, the heart responds by making lots of collagen and fibrotic tissue to try and make a Band-Aid, and usually goes overboard and creates too much, and that causes problems. So we expected, if anything, that we would get more collagen being formed, but in fact, we were surprised to find less. And we have some ideas why that might be, but we don't have a complete answer to it yet. So one thing we were able to do then was to remove these hearts, a pretty laborious procedure, remove the hearts and grind them up and look for gene expression, which allowed us to see what genes were being told to make proteins. And as we would have expected from our morphological studies, this gene expression profile that we could elucidate from our ground up hearts showed downregulation of many muscle proteins and collagens. In addition to that, though, we also found a very amazing and interesting result, which again, I'm still astounded by when I see it every time. And that is that something called a proteasome was significantly increased in terms of gene expression in these hearts. Now, proteasomes are essentially a cell's garbage disposer. So if you're a muscle cell, you make muscle proteins, they get put into the muscle myofibrils and they allow your muscles to contract and relax. But because they're working all the time, they get damaged. So it's really critical that the damaged protein molecules get pulled out, recycled through these proteasomes, and reused to make new, nicely functioning and shiny, clean muscle proteins. And what we found was that almost every single one of these many, many different subunits in these garbage disposers was dramatically upregulated. And when you see changes in gene expression in normal experiments, you get 3 or 4 different genes that are related to a function, you get all excited, oh yeah, this is upregulated. But to see every single one practically upregulated was just, wow, that was cool. And just to give you an idea of the fact that these really are garbage collectors, if you turn this protein on its end, there's actually a hole in the middle of this little mini garbage disposer machine, and that's where proteins get fed through, degraded, and then the pieces coming out get reused by the cell. So the cool thing about this really fun, exciting finding was we got to check it out on that next SpaceX 11 launch with our shiny new revamped VFBs. So we repeated the experiment. As I showed you, we sent a whole bunch more flies up. And when they came back, we wanted to find out, were there more protein aggregates created in space? And were there more of these actual proteasomes? We saw an increase in gene expression, but we had not seen, we couldn't tell if that translated to more proteins. So what we did was we sent up what we call a GFP tagged protein. GFP is green fluorescent protein. It comes from jellyfish. You may have heard about it. It also has garnered a Nobel Prize for its discoverers. And it's used to tag all kinds of things in cells so you can follow where they're going. So we tagged some proteins to see if we could identify clumps of unfolded, misregulated, undigested protein. And you can see in ground control hearts, there's a bunch of these green spots that are probably muscle proteins that have coagulated. So we could tell that at baseline, there are already some in flies. And then we stained those same hearts with an antibody that recognizes proteasomes. And here you can see the result of that staining in the same heart. And you can also see there's nice proteasome clumps that you would expect. And if you put them together, you can see that there's a bunch of these protein, we call them amyloid plaques, like occur in Alzheimer's disease, that co-localize with proteasome clumps, and you can see that down here. So then the question is, what happens in space? Well, as we predicted, there's a huge increase in clumped proteins, both in terms of numbers and size of these particles, but there's also this background, which is probably proteins that haven't yet come together to form a plaque. But we definitely could detect and quantify a big increase. And we also saw a huge increase in these proteasome particles. So that confirmed for us that the increase in gene expression we saw was now translating into an increase in production of proteasomes. And again, we saw them co-localizing, and often around nuclei, which are in blue here, which suggests that this is a process going on with new proteins that are being coded for in the nucleus and made as codes, the RNA comes out. So basically, our studies told us that in hearts, microgravity causes these garbage disposers to become clogged, because in studies on Earth, people have shown that when you get these large deposits of unfolded protein, the net result is to get into that hole that's in the middle of these proteasomes and basically clog up the works, like a garbage disposer if you put too much stuff in. And probably those unfolded proteins, those undigested proteins, are coming from this reorganization of the heart muscle that we see in the fly. Muscle in humans is probably too dense a tissue and too complex for us to be able to detect it as such in this kind of way in humans, but this suggests that it's probably happening also in humans. And we see this significant decrease in cardiac fibrosis. So quite a number of different and potentially informative pieces of information have come out of this study. In addition, though, we also looked at a couple other organs within the fly. Since we had the tissue there, we looked at the equivalent of your skeletal muscle, which is controlling the leg muscles and the wings of the fly. And we saw some very interesting changes here as well. Here, what you're looking at are in light or sort of medium gray muscle fibers in an electron micrograph. And you can see they're present in both ground control and space-flown flies. And you're seeing in between them mitochondria. So mitochondria are a powerhouse of the cell. They're an efficient producer of energy that is clearly needed for muscle contraction. And that's why they're situated in between these muscle fibers. These fibers are thicker than the ones that I showed you in the heart, but the organization is pretty much similar. And what we saw was a huge expansion of mitochondria between these muscle fibers in the heart, I'm sorry, in the thorax of the animal compared to ground control. Now, our first thought was they're trying desperately to make more energy, but our subsequent genetic analyses showed that that expression of mitochondrial genes was quite downregulated in these animals, and expression of what we call glycolytic genes was upregulated. And that's a process that's much less, it doesn't occur in mitochondria, and it's much less efficient. So it suggests that these mitochondria were becoming dysfunctional and perhaps not being effectively degraded as a part or cause of that dysfunction. So we have yet another clue about why skeletal muscle in both flies and astronauts is weakened after spending a significant amount of time in space. We also were able to look at brain tissue. So the interesting thing about a fly is that in the head, it has pretty much nothing but brain neural cells. And you can see that in this electron micrograph below the picture of the fly head. And so one of the reasons we wanted to look at brain tissue, in addition to being able to compare it to muscle tissue, was that in astronauts, when they spend even just a few months on the ISS, there are changes to their eyes. And it appears to occur predominantly in men compared to women, although there haven't been that many women, so that may be just a statistical sampling error. But essentially what happens is men in their fit astronauts, men in their 30s, go up to the ISS, spend 6 months there, and come back and they need reading glasses. And it's been documented to the point where it's a pretty well accepted phenomenon at this point. And the prevailing theory is that it's the change in gravity gradient when you're in space. Astronauts liken being on the ISS to standing on your head for 6 months because the pressure differential is equalized over your whole body. There's no gravity pulling your blood down. And so there's a change in the hydrostatic pressure in the eyeball that they think causes the change in astronaut vision. However, if it only happens in men and not in women, that might be a clue that maybe it's not the whole answer. So we were curious to see what was going on in neural tissue. And because the neural tissue, the brain, is a bigger organ than the heart, we were able to do something we couldn't do with the heart tissue, and that was to be able to look at both protein and gene expression from the same heads and be able to compare, okay, here's the genes that are changing. Is that reflected at the level of the proteins we can identify? And so we took the flies that came back, and in addition to looking at their heart function, when we dissected them, we took their heads and put them in test tubes, froze them, and put them away till we were able to later analyze them. And when we did our analysis, we found a whole bunch of gene expression changes, just like in the muscle tissue, where we had about 233 genes upregulated and 310 downregulated. And more protein, we detected more proteins that were upregulated than downregulated, but nevertheless, a significant number of changed gene and protein expression. What surprised us was when we looked for the overlap between the genes and the proteins they code for, we didn't find any. We found very few. And it turns out that this is not, we aren't the only ones who've seen this happen. So what this tells us at some level is that protein turnover, that proteasome function that we documented changing in the flies, may be a key to regulating some of the major changes that microgravity causes on organ systems in animals. So we went and looked at, in more detail, particular proteins that we had identified in the brain and at their gene expression. And to our surprise, unlike in the heart, where we saw upregulation of the genes and proteins for proteasomes in the heart, in the brain, we saw downregulation of gene expression and proteins. So it suggests that in addition to regulating protein, we call it proteostasis, regulating protein turnover, in general, it may be that it's something that's quite tissue specific, and we have to be aware of that when we start trying to devise countermeasures to help protect astronauts' muscles as well as eyes and brain. So what we saw in the brain, in addition to proteasome downregulation, was also quite interesting. And that is, we saw visual transduction genes being downregulated. Now, as I said before, astronauts have this problem with their eyes that they think is a result of hydrostatic pressure differences. But in the fly brain, there is no vitreous humor. There's no hydrostatic anything in the fly eye. It's a pretty almost crystalline structure. So it can't be why there's a change in eye gene expression. And so this is perhaps a clue for NASA that they need to look for more than just hydrostatic changes if they want to figure out what's going on in the eye problems that astronauts have. We also saw something that was kind of interesting. We saw changes in downregulation of circadian rhythm genes. And this has been seen in a whole bunch of animals, not just flies, in humans, in mice, in rats. And we're not really sure why, because they are in a light-controlled environment. So they're getting all their usual circadian cues, which tell them, you know, when to eat, when to sleep, just like they would on Earth. But there's something about the microgravity environment that causes changes to our circadian rhythm system that we can also, I think, we should also pay more attention to and spend more time studying. And we also saw, as I mentioned when I talked about the mitochondria, we saw changes in metabolic enzymes that were consistent with dysfunctional mitochondria and change in energy source to using glycolysis, a more inefficient way to produce energy. And we also saw immune system gene dysregulation, which again is something that has been seen time and again in all the different systems that have been investigated. So basically, in some of the things we've confirmed the utility of using the fly. We see similar changes that we see in other model systems and in humans, but it has given us some unique insights into particular molecular mechanisms that could be looked at in greater detail in other organisms, but is tough to do if you don't know where to look to begin with. So I want to sum just by kind of putting together the different multi-organ effects of weightlessness that we've been able to document. We have shown changes in cardiac size and contractility. And interestingly, we've recently analyzed flies that were born on the ISS, but allowed to become adults and grow up as adults on Earth. And some of those changes reverted. Not completely, not the contractility, but the heart size and some of the other protein organizations of the heart muscle rebounded after they came back. So that's encouraging news. We saw this surprising reduction in cardiac fibrosis, something that we need to explore further. Lots of decreased gene expression of muscle proteins, and defects in the protein recycling in both nerve and muscle. So we don't know, even though the effects were opposite, we don't know what the impact is on cell function when you increase or decrease proteasome function. So that's something we can further study on Earth. And as I said, it gives us clues as to how to prevent muscle loss in space and clues that may help us figure out why astronauts have altered vision. And of course, I need to thank the people involved. Now, this is just a fraction of the huge crew of graduate students and undergraduates and technicians and people at NASA who helped us pull these experiments off. But in particular, Sharmila Bhattacharya, who's at the time was an Ames research scientist and is now NASA's head of biological research, was our anchor to help us navigate through the complexity of NASA and NASA speak. And some researchers at UCSD and Ohio State University who also were helpful in getting us all together. And then a couple people in my lab who were on site with me down at Kennedy Space Center, including my technician, Erica, and key people from my collaborator, Ralph Bodmer's lab, who have done yeoman's work doing this genetic analysis and proteomics analysis. And of course, my sponsors, which include NASA and SBP, who were the key sources of funding beyond that initial prize from Space Florida. And I thank you for your attention, and I look forward to answering questions.
Thank you so much. We actually do have a ton of questions in the chat, so I will start.
Okay.
I will start with a question that came in actually by email. It was by Nina, and she wanted to know, are there any other aging lessons learned, like telomeres, if fruit flies have them, or brain size after space travel?
Yeah, so that's a great question. In Scott Kelly and his twin brother Mark, they did a telomere analysis. I think it was done at UCSD, and they found that actually astronaut Scott Kelly's telomeres lengthened. Now, telomeres are thought to be like a fuse, and they shorten as you age until there's no more telomere left, and then you start to get genetic damage. So lengthening of telomeres was a surprise. That's sort of like putting out the burning fuse and adding a longer one. It turns out it only lasted a short period of time. Again, we don't totally understand what's going on there with telomeres anyway, but it was a very interesting and not predicted thing that happened when they looked at humans. Flies have telomeres. We didn't have the money or resources to specifically look at them. So I can't tell you what happens in flies, but that would certainly be a very interesting study to do.
Maybe next time.
Hopefully, yes.
Another question that came in was, do any of your findings give insight into heart and brain function for people on Earth who don't travel to space?
Yes, they do actually. So many of the things that I described for the flies in weightlessness, the change in heart size, the remodeling, the contractility defects, those happen to people who are confined to bed, who can't, or who are convalescing or can't get out of bed for long periods of time. And so understanding, and in fact, bed rest is often used as a surrogate for weightlessness on Earth, although I'm not sure it's the best one to use. But I think understanding the fundamental cellular things that are going on in either of those cases will give us important insight. Each can inform the other. I think proteasomes are something we need to pay more attention to, for example, in people who are confined to bed. Is there some way we can help them work better, or not get gummed up and cause all this problem with protein function?
Another question is, why do the changes not revert to normal once the astronauts return back to Earth, or the fruit flies?
Well, it appears that they do slowly. There are some effects that we still are trying to document, and it's a little harder with humans for a bunch of reasons, but not the least is that astronauts who come back do not want to share their medical records, because if there is anything that doesn't get better, that would be grounds for keeping them on the ground and not letting them have additional missions. So their records are jealously guarded. In fact, I think Scott and Mark Kelly are the only two astronauts who have given mostly open access to the results of their studies. But it looks like from the fruit fly studies, our recent analysis, that there is a fair amount of recovery, at least in terms of the overall organization of the fly heart that we see after the flies have come back, although it does appear that their heart contractility is still compromised. And that's certainly something to be concerned about. But keep in mind, human hearts are much more robust and much thicker and have multiple tissue layers. So small changes in heart cell function might not have as much of an impact as it does on the fly heart.
To follow that, someone asked, hasn't permanent osteoporotic changes in astronauts been documented?
Yeah, that's absolutely the case. It's a big problem. And there are several mouse studies that have been designed to look at that. There unfortunately is no bone that we can look at in the fruit fly, so that's not something that we can use as a model for trying to mitigate effects of space flight. But there have been some efforts to reverse that have been successful, at least in mice and rat studies. And that is, of course, directly applicable to humans, although it's still completely experimental at this point.
Okay. Have there been any variations in traits past the line of flies that were born in space versus those that were not born in space?
Yeah, we haven't had the resources to follow them for generations after they came back. We just looked at the sort of immediate recovery. I would be surprised if particular genetic traits were changed, but what could change and is actually likely to change are things called epigenetic traits. So these are changes that occur to the DNA that don't involve changing the genetic information, but change how it's expressed, how it's translated into functional proteins. And that would be a fantastic study that the fruit fly would be very well suited for. And there's plenty of good questions that we would be able to ask if given the chance.
Okay, a couple more. How much of the changes seen are due to radiation in the fruit flies and the astronauts?
Yeah, that's also a really good question. And that's something we're going to have to, we're thinking of ways to propose to do on the moon, because ISS is still within the Earth's magnetosphere. And so it's still protected to some extent from the ionizing radiation that is certainly going to be deleterious to humans. In fact, that, in my opinion, is the biggest problem NASA has to face. And that wouldn't be the case on the moon. So we're already trying to put together studies to take flies to the gateway when it's finally created. I don't think we even need to go down to the moon surface to do those kinds of tests, because the gateway, the orbiting platform would be within, would not be protected by any magnetosphere or any other protective force. And that's something I would like to do, and we're thinking about ways to do it.
Sounds like you have a ton of research left.
Oh yeah, I have so much data that I'm still analyzing from those flights. It was a massive data collection experience, and we're still combing through it to get new clues. It'll take us a long time to finish.
So the same person that asked the question about the radiation made a comment saying that they were surprised that astronauts are not required to share their medical data. Seems like something required to release for humanity's good. So.
Yeah, it's quite controversial. And as I said, the astronauts jealously guard it because they don't want to be grounded. And I guess I understand that. But yeah, that's NASA. They are very strict about not sharing private medical information. And that's what made the twin studies so remarkable, was they agreed to have their private medical information shared to some extent. Even they didn't share it completely, but to some extent it was. And so I think that's just points to the utility of model systems to get at some of these questions, since it's tough to invade an astronaut's privacy. And in addition, every astronaut's genetic makeup is vastly different from every other astronaut. So you don't really know what genes are that might make a difference in one astronaut's response to microgravity compared to another. Whereas if you've got mice, rats, or fruit flies, their genetic backgrounds are typically identical. So any changes will immediately be visible, be evident. Whereas if you compare one human to another, yeah, we don't have the bioinformatics capabilities at this point to really be able to tease that apart. Maybe in 10 or 15 years, but not now.
Okay, so I have a couple more questions for you, and people are still putting things into the chat. I am going to circle back to the beginning of your talk when you were talking about the commercialism on ISS. And so with the rise of the space tourism idea, would you say that leisure space travel is safe for humans?
Right now, what I believe it's Bezos's Blue Origins are designing a rocket that is going to take tourists up for a brief flight of a few hours, I think, not more, but into the zone where the ISS is. And almost certainly that would not pose an immediate threat to humans, that brief and exposure to microgravity. You need to probably be in good physical health to begin with, but I don't think that kind of travel would be an issue. What will be an issue is if they want to go to the moon and stay on the moon for a week or a month and then come back. Then you probably need to start worrying about more long-term effects, and especially then ionizing radiation.
So one of the comments you made is the 50% of the activity on the International Space Station is commercial. What's considered commercial?
They have so many things going on. One, so these are experiments that are sent up by commercial enterprises. So they'll make crystals in space, because without gravity, you can make much purer crystal structures to do atomic, to look at the atomic organization of proteins, for example. You can make different drugs more pure. They end up being made in a purer fashion on the ISS in weightlessness. There was one experiment that went up when I was sending up our flies that basically took out, you know, those bracelets that are kind of a piece of metal that slaps around your wrist and just by tension kind of coils. They had a solar panel that was initially coiled like those bracelets, and they sent it up to have it tested to see if they could deploy it like you would expect one of those bracelets when they straighten out and become stiff and straight from the ISS, and that was a successful experiment. As I said, there's experiments on osteoporosis that have been done. They look at plant growth. They want to understand what it is about gravity that causes plants to send their roots down. What is it that plants are actually sensing? And so there's all those kinds of experiments that will be needed if we're going to be growing plants on the moon, for example, but also you can imagine trying to make roots grow deeper if water table lowers and things like that. If we know how to manipulate that response to gravity within the plants, then perhaps we can take better advantage of what water is available.
Okay, given that we share roughly 75% of disease-causing genes with the fruit fly, were you able to determine any other effects from space travel on humans from those on the heart?
Yeah, we actually have some interesting proteins that we identified in the heart that have not been shown yet to be functional in human heart. And these are proteins that bind to structural proteins in muscle cells and sense tension from outside the cell. And so it's part of a feedback system that helps your heart know how much to contract and how forceful it needs to be. And we've identified a protein that's not been shown yet to be functional in human heart. And we're currently studying it in the fly to see if it does what we think it does And we'll go from there to our other model systems, for example, the zebrafish heart. And then we can go to human pluripotent stem cell derived cardiomyocytes to see if those genes have a function in human heart cells. So stay tuned.
Well, thank you. Okay, I have a question from a while ago. I have to go back. The question I guess is, who oversaw the box of your fruit flies in space? Was that, who was
that?
Was
me. That was me and Sharmila, the first box. And we had one of Sharmila's technicians, the three of us went to Kennedy Space Center, spent a month there. I finally had to come back. I'd been over a month and there's always rain delays. I mean, to put a space launch station in Florida, which has the most lightning strikes of any state in the entire country, and then only allow launches if there hasn't been a lightning strike for 30 minutes, means that your flight often gets delayed. And so after like 3 weeks or 4 weeks of delay after delay after delay, I finally said, all right, you guys, I got to go back. I got a lab to run. And so actually Sharmila did too. She went back to Ames and we left our technician there to oversee the final loading of the boxes. But for the second mission, when we put up all of those 9 boxes, we worked literally day and night through cancellation after cancellation, because we had to regenerate our samples every time it was canceled, and refill boxes. And so we just spent, I think, 3 weeks of working 12-hour days, generating new samples every day until it finally lifted off. And then we could go home because they splashed down off Los Angeles, off Long Beach, and so weren't far from us. We got them pretty quick after that.
So I guess the question is, you guys do all the prep and then it goes on to the vehicle, and then is it basically it just sits on there and it does all its work?
Yeah, that was the beauty of it. And that's one of the reasons that we got picked, because it required no astronaut time except to take them off the capsule, the Dragon capsule, and stick them into a shelf or an incubator. And the reason that's so key is apparently the astronauts are also working 18-hour days, just trying to get all the experiments done and the maintenance done that they have to do on the ISS. So their time is scheduled to the minute every single day aboard the ISS. And they have to do 1.5 to 2 hours of exercise to try and maintain muscle mass, which I should have mentioned helps, but doesn't prevent loss of muscle. So they're very regimented and their time is really precious. And so not to have to use astronauts was a huge advantage.
Thank you. We do have one question, but it's very, very far off the topic. So I'm going to ask you and you can decline from response. And I'm going to ask if there's any last questions to throw them into the chat. So this is a follow-up to some of the questions about the astronauts, with a question about embryos in space and whether or not there's, of an embryo being made in space. The person seems to think that a sperm and an egg would have a hard time getting together in space.
Well, they didn't in the fruit flies because they were totally able to reproduce quite nicely in space. It didn't seem to be a problem. And part of that is because sperm and eggs are in a liquid environment. It's not like they have to walk or, fly or anything to get to each other. So that really isn't a problem. What we were curious about was whether embryological development would be altered by microgravity. And our recovery experiments where we took eggs laid in space, and when they came back, we waited until they became adults on Earth and then analyzed the adults. Those hearts looked not too terrible. There were some, as I said, contractility problems, but they still functioned better than the space-flown adults. So it would suggest that embryological development isn't severely compromised and that the problems come when you start trying to make a mature heart muscle and you can't turn over the protein properly, or you can't maintain that mature heart muscle. So that's a hint that it's probably not embryological development.
Well, thank you. And I wanted to thank you for participating tonight. And I wanted to thank our audience for joining us tonight for our DMF talk. And
thank you, everyone. It was great. It was a pleasure to tell you all about our fun experiences and kind of, I think, pretty exciting results that we actually will be following up on Earth, because it's given us some really cool new insights, I think.
well, thank you. Okay, everyone, have a great night.
Thank you all. Have a great evening.